So we are back with the second lesson in Unit 4. Today's topic is mitosis and cytokinesis. I mentioned both of those in the previous lesson, but we're really going to zoom in on exactly what happens during each phase of mitosis. And also we'll take a closer look at the process of cytokinesis, the actual physical splitting of a cell to make two cells. I'll be discussing content that is covered on pages 185 to 190 in the Campbell Biology in Focus textbook. So I mentioned in the previous lesson that there are a number of terms in this unit that sound alike, and this can be potentially confusing for students. So chromatin, chromosomes, and chromatids. These are all related terms, but they do have different meanings. So not shown in this image that you're currently looking at on the screen is chromatin, T-I-N, chromatin. Chromatin refers to long, thin, relatively uncoiled or loosely coiled strands of DNA. And that is the nature of DNA most of the time. If you look at the nucleus of a cell under a microscope during interphase, G1, S, or G2, and remember, interphase is most of the life of a cell. During interphase, you will not see chromosomes. You cannot see discernible individual chromosomes. It's impossible to count the chromosomes in the cell because they simply do not exist yet as chromosomes, but rather the DNA exists as kind of a loose, dispersed, stringy mass, okay? And it's only during mitosis that those individual threads of chromatin will coil and shorten and thicken and twist up into tiny packets of DNA, which we refer to as chromosomes. And at that point, they become visible with the microscope, and we can actually count the number of chromosomes that we see in each cell. Now, in this image, you're looking at two chromosomes. One has been color-coded blue because it was inherited from the sperm cell, and the other is color-coded red because it was inherited from the egg, and that's just a convenient way to color code them. But notice these chromosomes have not yet been duplicated. And what that means is that each chromosome only has one copy of itself. Each chromosome at this point only has one chromatid. Now that will change once we get past the S phase. After the DNA is duplicated, these chromosomes will look quite different. But right now, this is one chromosome. It has one chromatid, or one copy of itself. And then over here is a separate chromosome, and it too has one chromatid, or one copy of itself. And if you could see chromosomes during G1, this is how they would look. You actually cannot see chromosomes during G1 because remember, the DNA does not coil up and actually form chromosomes until mitosis begins. But let's look. So here's the chromosomes I just showed you, and here's the same two chromosomes after they have been duplicated. This is still two chromosomes. But now each chromosome has two identical copies of itself because we're looking at the chromosomes after the S phase. The two identical copies that make up this chromosome are called sister chromatids. You can see I labeled them over here. So sister chromatids are the two identical copies that make up a duplicated chromosome. So a chromosome can have one chromatid, as you see in this picture, or a chromosome can have two chromatids. And we are now looking at two duplicated chromosomes, each chromosome having two identical copies, two sister chromatids. The area of DNA that kind of joins the two chromatids together is called the centromere. And you can't see it in this picture, but on either side of the centromere, like right here, and then on the other side right here, there's a special protein called the kinetochore. We'll talk more about that later, but the kinetochore is the attachment site for spindle fibers. Basically, tow cables are going to attach to those kinetochores on either side of the centromere, and that is going to allow the chromosomes to be moved around and eventually pulled apart. We're going to separate the chromatids during mitosis. Whenever you have a pair of chromosomes that are the same size and the same shape, and they carry genes for similar traits, like maybe this is a gene for height, and this is a gene for height. Maybe this is a gene for eye color, and so this would be a gene for eye color as well. We call chromosomes like that homologous. So this would be referred to as a homologous pair of chromosomes. And the homologous chromosomes are not identical. I repeat, they are not identical to each other. Because remember, you inherited one of these chromosomes from your dad, and the other one you inherited from your mom. So of course they're not going to be identical. This one might have a gene for tall, and this might have a gene for short. This might be blue eyes, this might be brown eyes. But they are homologous because they're the same size and the same shape, and they carry genes for the same kinds of traits. All right, so I just wanted to make sure. I know that's not terribly difficult thing to grasp, but students do sometimes get confused about the difference between chromosomes and chromatids. So I'd like to call your attention to this first cell in this image. We're looking at a cell that's still in interphase. This is G2. So we're getting pretty close to the start of mitosis. Remember, G2 is when the cell is preparing for mitosis. You can see that the very beginnings of spindle fibers have started to form in this cell. Whenever the centriole pairs have little beginnings of spindle fibers forming around them, we refer to this as an aster, which literally means star, because it does kind of look like two twinkling stars that are sitting right beside each other. But those are microtubules. Those are special proteins that are going to be used to build the spindle once we begin the process of mitosis a little bit later. So that's what we mean when we say the cell is preparing for mitosis. It's starting to accumulate some of those microtubules that will be used for the spindle. Now you can always tell when you look at a cell if it is in interphase, because look in the nucleus. We don't see chromosomes. If I was to ask you to count how many chromosomes are in this cell, or how many pieces of DNA, you could not possibly count. It looks like a plate of spaghetti. The DNA is so dispersed, and it's made up of these long, thin strands of chromatin. Now, the DNA has been duplicated at this point, and I know that, well, first of all, it tells me right here, but I know that the DNA has already been duplicated because, look, we're in G2. And remember, we had G1 when the cell grew larger, and then we've already had the S phase where the DNA was copied. So there's now twice as much DNA inside this cell as there was originally. The nucleolus is still easily visible. That's the dark spot where ribosomal subunits are made. Notice the nuclear envelope is fully intact. Let's move over to prophase. This is now the beginning of mitosis. Once we get to this picture right here, mitosis has begun. This cell has now begun the process of dividing its nucleus to make two separate, genetically identical nuclei. Remember, that's the goal of mitosis. That is the purpose of mitosis, is to take all of the DNA in this nucleus. And we know we have two copies of every chromosome because we already made the copies back during the S phase. So our purpose in mitosis now is going to be to separate those copies from each other and split the nucleus to make two separate, genetically identical nuclei. Okay, so let's discuss the changes that take place in the cell during prophase. First of all, you see the centrosomes, which includes a pair of centrioles. We have two centrosomes, and they started out side by side. But notice in prophase of mitosis, they begin to migrate apart. And they get further and further apart until at the end of prophase, they're almost completely at opposite ends of the cell. Between those migrating centrosomes, we have this network of microtubule fibers that are starting to form. We refer to that as the spindle. These are spindle fibers, and they're eventually going to, some of them at least, are going to connect with the chromosomes and start attaching to them like tow cables. During prophase, the long, thin strands of chromatin are coiling and shortening and thickening to form distinct chromosomes. And by the end of prophase, we can really see those chromosomes as individual chromosomes, and we can count them: 1, 2, 3, 4. They're only showing 4 chromosomes in this picture for simplicity, but if this was a human cell, there would actually be 46. Notice the nuclear envelope is breaking up because, frankly, it's just in the way. In order to move the chromosomes around, we need that nuclear envelope to break down, and that's what it's doing. The nucleolus temporarily disbands, and all of the RNA within the nucleolus scatters throughout the cell. And so notice by the end of prophase, we do not see any remnants of that nucleolus. Now, in your textbook, these two images are labeled a little bit differently. I think this picture is labeled as the prophase picture, and I think probably your textbook would call this pro-metaphase, which to me is a little bit silly. I prefer to think of this as early prophase and late prophase, and I think that's how most biologists would sort of think of this. I mean, all of these phases are arbitrary. We name the phases just so it's easier for us to keep track of the flow of events. But we're going to consider both of these pictures to be prophase. We're going to call this early prophase, and then this would be late prophase. And there's a lot happening during prophase. So I made a little list, and as you take notes, make sure you write down what happens during each phase. That's going to be really important, that you keep up with what happens during each phase, and also that you recognize what the cell looks like during each phase. So in prophase, the chromatin coils up or condenses to form chromosomes. The nucleolus disappears, the spindle fibers form, the centrosomes migrate apart. Remember, they started out side by side, and they get further apart. By the end of prophase, they are just about on opposite ends of the cell. The nuclear envelope breaks down, and here's a fancy term, the kinetochore fibers attach to the kinetochores. So what in the world is that talking about? I've got a close-up picture of that that I can show you if I can find the right. Bear with me here. Okay, so here's an extreme close-up, and you can see the microtubules which make up the spindle fibers attaching to this kinetochore on this side of the centromere, and then on the other side of the centromere we have another set of microtubules attaching to this kinetochore. So basically, by the end of prophase, or certainly by the beginning of metaphase, every single chromosome is going to have a spindle fiber attached to one side and another spindle fiber attached to the other side. It's basically tow cables that are going to try to pull that chromosome in two separate directions at the same time. And we'll see how that turns out in just a minute. So here's pretty cool photographs that were taken with an electron microscope, color-enhanced photographs, and we can see the centrosomes and the early formation of the spindle fibers. Notice the nucleus is still pretty much intact, but by the end of prophase, we no longer have a nuclear envelope. We have centrosomes that have now migrated to opposite ends of the cell. The next phase of mitosis is metaphase. It's the most easily recognizable of the mitosis phases because all of the chromosomes are aligned within the same plane, and we sometimes refer to this as chromosomes lining up along the equator. Kind of think of an imaginary line running through the middle of the cell. That's not necessarily the best term to use because sometimes the chromosomes will line up vertically instead of horizontally. So just imagine if the centrosomes were on this side and this side, then the chromosomes would be lined up in a vertical orientation. So it may be confusing to call this the equator. I notice your textbook refers to it as the metaphase plate. So for your notes on metaphase, the centrosomes reach opposite poles. Remember, they were almost completely at opposite ends by the end of prophase, and now they truly are on completely opposite ends of the cell, and the chromosomes align along the metaphase plate, or the equator, if you want to call it that. And the reason they line up like this is because every single chromosome has a spindle fiber that's trying to pull it down, and another spindle fiber that's trying to pull it the other way. One spindle fiber is trying to pull it to this end. The other spindle fiber is trying to pull it in the opposite direction. So basically it's a tug of war where every chromosome is being pulled in two directions at once. And so where do they go? They just go to the middle. Until eventually there's enough force being applied by these opposing spindle fibers that the centromere right in the middle of the chromosome is going to split. Every single chromosome is going to split in anaphase so that the two identical copies of each chromosome can be pulled apart and can be separated. So I don't want to get ahead of myself. Let's make sure we understand metaphase first. Chromosomes lining up single file along the middle of the cell. And you notice when metaphase occurs in a real cell, there's so many chromosomes, it's hard for them to get in an exactly straight line. So you kind of have to use your imagination to see that the chromosomes are in a line. And then anaphase happens. Anaphase begins when each chromosome splits in half. And what we used to call sister chromatids, when they were attached, see, two sister chromatids making up this chromosome. Well, as soon as they split apart, as soon as this one starts being pulled this way and the other copy starts being pulled the other way, we don't call them sister chromatids anymore. We call them daughter chromosomes. The name changes to daughter chromosomes. So this is anaphase. The centromere, the middle part of every chromosome, splits in half. The spindle fibers shorten, and that's actually a very complicated process that involves motor proteins and different enzymes, and scientists are still kind of trying to figure out how this mechanism works that actually allows the spindle fibers to get shorter and shorter. But a simple way to think of it is that the spindle fibers are like fishing line, and it's like one fisherman is reeling in this copy and pulling it to this riverbank, and at the same time a fisherman on the other side of the river is reeling in his line and pulling the other copy to the opposite riverbank. Every single chromosome, remember, has two copies. So this is a complete copy of the chromosome. Here is another complete copy of that same chromosome. And so we want one copy of each chromosome to be pulled up to this end, and we want an identical copy of each chromosome to be pulled to the other end. That's the whole point of anaphase. That's the whole point of mitosis. So the sister chromatids, which we no longer call chromatids, they are now daughter chromosomes, begin moving toward opposite ends of the cell. This is also the point where the cell starts to become a little bit more elongated. You see how in this picture in metaphase the whole cell kind of looks round, and then in anaphase it starts to take on a little bit of an oblong appearance. That's because these spindle fibers, some of them don't touch the chromosomes. Sometimes you have a spindle fiber that goes all the way to the middle of the cell, and it kind of touches another spindle fiber that's coming from the other end of the cell, and they kind of push. Instead of pulling, they push against each other. So it's kind of the opposite of a tug of war. And they're called non-kinetochore microtubules. These are the spindle fibers that do not connect with chromosomes. Their job is to push against each other and make that cell elongate. Anaphase is also interesting because of a little technicality that affects the number of chromosomes. Let me go back to the picture. So if I was to ask you to look at the metaphase cell and count how many chromosomes you can see, that would be 1, 2, 3, 4. But if I told you to look at anaphase, the very same cell, just a few moments later, now how many chromosomes do you see? 1, 2, 3, 4, 5, 6, 7, 8. The number of chromosomes has doubled. And I know that's confusing because the total amount of DNA is the same. If we were to count chromatids, the number hasn't changed. 1, 2, 3, 4, 5, 6, 7, 8 chromatids. We still have 8 chromatids in this picture. But remember, when you have sister chromatids that are connected together and they haven't separated, then we call this whole thing a chromosome. As long as the two chromatids are still together, it's a chromosome. But as soon as they separate and those chromatids start moving apart, each chromatid becomes a daughter chromosome. So the number of chromosomes actually doubles in anaphase, and these 4 chromosomes will end up in this nucleus. The other 4 chromosomes will end up in a separate nucleus at the other end of the cell. When the cell splits, the number of chromosomes per cell will go back to the original number, which in this case will be 4. So just to be clear, if this was a human cell, there would be 46 chromosomes in a straight line in metaphase. So if this was a real human cell, how many chromosomes would we have in the anaphase cell? If you said 92, you are correct. There would be 46 chromosomes being pulled to the north end, and another 46 identically the same chromosomes, identical, another 46 being pulled to the south. 46 chromosomes going north, 46 chromosomes going south, and that's how we're going to end up with two separate cells, and they're each going to have 46 chromosomes. Nice little picture of anaphase. You can start to see a little bit of empty space right in this area because half of the, I shouldn't say it that way, one copy of every chromosome is being pulled to the north, one copy of every chromosome is being pulled to the south. Okay, after anaphase comes telophase, and many of the events that happen during telophase are the opposite of what happens in prophase. So in prophase, the spindle fibers formed. In telophase, the spindle fibers are breaking apart and disappearing. They've already done their job. The job of the spindle fibers was to pull the chromosome copies apart, and that's already happened. So spindle fibers are breaking down in telophase. In prophase, the nuclear envelope disintegrated. Now a brand new nuclear envelope is forming at this end of the cell and at this end of the cell. In prophase, the nucleolus disappeared. Now it is reappearing twice, once at either end of the cell. In prophase, the chromatin coiled up and formed chromosomes. Now, in telophase, what's happening? All the chromosomes are uncoiling. They're once again becoming long, stringy, thin strands of DNA. Once again, it becomes difficult to count how many pieces of DNA are in this nucleus. So we go back to chromatin. So let's just list the telophase events. Daughter chromosomes reach opposite poles. A nuclear envelope forms at each pole. Each end of the cell is going to have its own nucleus now. Chromosomes uncoil back into chromatin. The nucleolus reforms within each nucleus. So remember, we're going to have nucleolus at this end and a nucleolus at this end. Spindle fibers break down. And notice how I worded this. Cytokinesis, the physical splitting of the whole cell, usually begins during telophase. I say usually. Sometimes in anaphase, you'll start to see a little bit of a folding in on either side, but it definitely becomes more pronounced. You see that this cell has started to pinch inward here and here. We call that pinching inward a cleavage furrow. This is an animal cell, and the way animal cells split is they pinch inward until that cleavage furrow gets deeper and deeper, and eventually it splits the cell right down the middle, and it'll actually separate and become two separate cells. That process of cytokinesis, the physical splitting of the cell, normally begins during telophase. Sometimes you might see a little hint of it in anaphase, so don't be confused by that. But typically we think of cytokinesis as mainly occurring during telophase. Okay, so let's think about what we have accomplished. After telophase is complete, we have a nucleus here and a nucleus here, and they are genetically identical. They have exactly the same DNA. Every piece of DNA in this nucleus has an exact copy in the other nucleus. That was the whole point, right? And after cytokinesis is complete, and this cell has split, This nucleus will be in one cell, and the other nucleus will be in a completely separate cell, because these two cells will cleave, they will separate, they will no longer be one cell. The final result of mitosis is two genetically identical cells, and both cells will have the exact same number of chromosomes. So if we started out, I'm just going to use humans as an example, if we started out in prophase with 46 chromosomes, now we can only see 4 in this picture, but let's just pretend there was 46 chromosomes in this picture, in this one cell. Then by the time we finish cytokinesis, there will be 46 chromosomes in this cell and 46 chromosomes in this cell. It's a really cool picture of, if I can find it, here we go. Really cool picture of telophase. We can see that the red area is the cytoplasm. So this used to be one cell, but you notice it's pinching in on either side. We have a nucleus here and a separate nucleus here, and eventually this cell is going to pinch all the way across this middle, and it's going to separate into two separate genetically identical cells. Now, one final word I want to mention here about cytokinesis. All of these pictures that I've shown you today have been animal cells undergoing mitosis. It's important to note that plant cells also do mitosis. Plant cells have to divide the same as animal cells, because plants have to grow and develop, and they have to make new cells and heal damaged tissue, and a lot of the same things that animals do. If we were looking at plant cells doing mitosis, one big difference is you would not have these little centrioles. You see this circular area where the spindle kind of forms out of. That circular area is called the centrosome. And in an animal cell, it always has a pair of centrioles. So there's a pair of centrioles at this end, and then we have another pair of centrioles down at the other end. Well, plant cells don't have centrioles. But plant cells do still form. We don't know exactly how this works, but somehow plant cells do form a spindle, and they use spindle fibers to pull the chromosome copies apart, same as an animal cell. So mitosis is pretty much the same in a plant cell as it is in an animal cell. But the big difference is cytokinesis. Because remember, if this was a plant cell, there would be a rigid outer wall made out of cellulose. There would be a cell wall around the outside, and that cell wall is rigid. It does not bend easily. So unlike the plasma membrane, which can bend and pinch in, a cell wall can't do that. So cytokinesis does work differently in plant cells and animal cells. So I just wanted to go through that quickly. So running around the inner surface of the plasma membrane in an animal cell is a contractile ring of protein microfilaments. Think of this as like a drawstring on a pair of jogging pants. It's actually made up of two different kinds of protein filaments. One is called actin and the other is called myosin. But it's essentially a drawstring that runs around the inner surface of the plasma membrane of an animal cell. And when it is time for cytokinesis to begin, that drawstring starts to pull in tighter. Just like if you were cinching down on a drawstring, it's going to make your jogging pants feel tighter. It's going to start to squeeze your waist. And when that happens to an animal cell, we see that cleavage furrow. And as that ring contracts tighter and tighter, the cleavage furrow gets deeper and deeper until the cell completely separates and cleaves into two separate cells. So actin and myosin proteins form a ring around the inside of the cell membrane. That ring contracts. That means it gets tighter, like pulling on a drawstring. That creates the cleavage furrow, and eventually it splits the cell into two separate cells. That's how animal cells do cytokinesis. But if you look at the plant cell, it's totally different. Let's assume mitosis is complete, so we've gotten all the way to telophase. So we have a nucleus on this side of the plant cell, we have a nucleus on the other side. Now it's time for the plant cell to do cytokinesis. Well, there's no drawstring, because remember, plants have a cell wall in addition to the plasma membrane, and I don't care how hard you pull on a drawstring, that cell wall is not going to bend. So what plant cells do is they use their Golgi apparatus to make lots and lots of tiny little vesicles, and each vesicle contains a tiny amount of cellulose, cell wall material. And as these vesicles are formed by the Golgi apparatus, they start to line up along the middle of the cell, kind of the same way the chromosomes lined up back in metaphase, but this time it's little vesicles containing cellulose. And as they line up and more vesicles arrive along this line, they start to smush together. They start to fuse together with one another, and all of that cellulose gets dumped and assembled into a growing wall. This is referred to as the cell plate at this point, but eventually this wall will grow all the way, it'll form all the way across, and it'll split that cell right down the middle. So plant cells, even after cytokinesis, after the splitting process is complete, they still remain connected. They're still going to share this wall in between. This would be kind of like if I brought in a bunch of blocks and I built a wall right across the middle of my classroom, so that half of my students were in the front room and the other half of my students were in a totally separate room, and we couldn't see each other because we're on opposite sides of a wall. That's what plant cells do. So plant cells do a process called cell plate formation, where cellulose-filled vesicles from the Golgi line up along the middle, forming a cell plate. Eventually, those vesicles fuse together, and the vesicles themselves actually form a new layer of cell membrane on either side of the wall, but the cellulose inside those vesicles fuses together to make a brand new wall. So I hope that has helped you understand mitosis and cytokinesis a little bit better. This is the end of Lesson 2. And I will see you real soon for Lesson 3. I hope you have an outstanding day. Take care.